And when we're looking at how this is inherited, you should be familiar with the Punnett square. We know that if there are two heterozygous parents, meaning each of them carries a recessive gene for the disease, that's going to make them a carrier. Okay, if they carry that recessive if they carry that recessive gene for a disease, they are a carrier of that disease. So we have a carrier mom and a carrier dad here, and we know that the only gametes that they can make are the ones that they possess. So they will make gametes with both the normal gene and the abnormal gene. In this case, we're talking about little a being the recessive gene for disease. Father, same thing. And we know from doing our Punnett squares and getting lots of practice on that, that we're going to have one normal child who doesn't even carry the gene, and then we'll have two carrier children, but we will have a 25% chance of having an affected child. An example of this is cystic fibrosis. And cystic fibrosis is the most common lethal genetic disease in Caucasians in the US. It's caused by mutation in the cystic fibrosis gene, and that affects cells in the exocrine glands. And exocrine glands are glands that secrete to surfaces. So they actually have like tubes that go from the gland out to the surface. The salivary gland, for example, is an exocrine gland because it secretes saliva into your mouth. So what happens is in cystic fibrosis, those secretions are very, very thick. So normally, we've talked before about mucus, for example, being on our surfaces in our respiratory tract. And it's there to collect dust and dirt and bacteria, things that we want to trap before they get into the lungs. And we know that our cilia on the surface of the cell brings up that mucus. It provides motility and movement for the mucus, and the mucus comes up to the back of our throat and we swallow it, unless we have a cold or something else that makes that more difficult. And then we might cough it up. But with cystic fibrosis, the mucus is so thick that it's very difficult for the cilia to move. And so that thick mucus ends up trapping things that we would like to get rid of. It leads to frequent lung infections in cystic fibrosis patients. So we know that that happens, but also it's we associate it with lung disease or pneumonias, and that is a very real problem, but it affects all the secretions. So it also is a digestive problem as well. When I started in the hospital way back when, it was unusual to see a cystic fibrosis patient live past 20 because of the infections that they encountered, particularly lung infections or pneumonia. Now they have much longer lifespan, but it's still usually too short. Another disease that is an autosomal recessive disease, meaning it's only going to be, the person's only going to get the disease if they get both defective genes. And that's Tay-Sachs, and this is a horrible disease, and unfortunately we have not made as much progress as we have with cystic fibrosis. Tay-Sachs is present. We've always known about Ashkenazi Jews having Tay-Sachs, but it's also present in Old Order Amish in Pennsylvania, and there's even some French Canadian communities. Frequently, these are small areas and small communities, and so it gets passed, the gene gets passed a little more easily, but Tay-Sachs causes an enzyme defect, and the result is accumulation of a lipid in the brain cells, and it starts out, the baby is normal and starts showing signs as developmental milestones are looked for at the doctors. the baby stops meeting those milestones and starts regressing, and usually dies by age 3. So what would we expect a pedigree analysis for someone, a family of cystic fibrosis patient to look like? And this would be any autosomal recessive. Okay, this would be a very similar picture. So we're talking about the mating of male and female here. At the time, they may not know that they carry this gene, because remember, they will appear normal. Their genotype will be abnormal, but their phenotype will be normal. But once they have children, you start to see of these children that a number of them, in this case 2, are affected. And we know that this is an autosomal recessive because it is not sex linked. Our choices are autosomal or sex linked. And we can have carriers of sex linked diseases, but they're always going to be female, right? A male can't be a carrier. He would actually have it if this was sex linked. So we know that this is an autosomally carried recessive disorder. Autosomal dominant disorders are different than the autosomal recessive disorders in that they're going to be expressed if the individual has a heterozygous genotype. So that means that males and females are equally affected, and that affected children have affected parents. So remember that whether the person has 2 of the genes or is heterozygous and only has 1 of those genes, they will show the traits of the disease. So they will have the phenotype. The example I give here is Huntington's disease. And Huntington's disease is a neurological disorder that leads to progressive degeneration in the brain. Basically, there is toxic byproducts that build up in the neuron, killing the neuron. And as you know from the tissue lab, the neuron is responsible for sending messages through the brain and spinal cord. Symptoms usually do not occur until 35 to 45 years old, and that is why Huntington's disease still exists. For many, many generations up to now, most people probably would have chosen, if they knew they were going to get Huntington's disease, maybe not to have children and not pass along the gene. But at 35 years old, many people have already had children. So by the time they presented with the disease, they've already passed on the gene, and that it's been in our gene population for years. So it's important to remember too that just recently, and I say recently, but really for the last maybe 30 years, we've been able to identify this gene in people. Okay, we weren't able always to do that. We could recognize the phenotypic effects of Huntington's disease, but we could not look at the gene and determine that someone had the gene before they had the symptoms. So actually, when I was in college, one of my best friends in college her mother had Huntington's disease, and by that point she was well into showing the symptoms, so they were aware that she had Huntington's, and she was really concerned because she and her brother then both had the chance to have Huntington's disease because it was a dominant disorder. So her father did not have it, but all they needed, either one of them, was one of the abnormal genes. And so when it first came out and we were in college, it may have been a little earlier than that, but they came out with the fact they could check her. They could test her and determine whether she carried the gene for Huntington's. And she was having a very hard time with this because she knew what was in store. She'd watched her mother going through the first stages of it. And she didn't know, first of all, whether she wanted to know. You know, if you think about it, would you want to know basically that you are going to start having brain degeneration and die? So she was debating that, but then she had a real issue too with autosomal dominant disorders are going to be expressed in 50%, and you can do the Punnett square, but in 50% of the patients. So if they're going to be expressed in 50% of the patients, she figured that by the numbers, either she or her brother would get it. And she felt as if she determined that she didn't have it, then she basically would be telling her brother that he did have it. She did eventually decide to have it done, and she did not carry it. I don't know what happened with her brother, but certainly led to an ethical dilemma for her. So here is a pedigree analysis again. In this case, remember we're talking about an autosomal dominant. So the parent will have and will express that abnormal gene. So in this case, we see the mother, and they show 4 children, and 2 of those children carry the Huntington's disease. The child that did not carry the Huntington's disease from the mom, when he mated, their child does not show it because there is no Huntington's gene. All right. Over here though, I'm sorry, you cannot see that yet. All right, so over here, this child does not carry the gene at all. So it doesn't matter, he can have multiple children and they will not have Huntington's. But this child carried the gene, got it from the mother, passed it along, and married. And once again, you'd be talking about the probability of about 50% of the children having the gene. So the way you could tell this was an autosomal dominant, first of all, the mother carried it and had the disease as well, passed it on to about 50% of her children. It doesn't matter whether they were male or female. They're going to be affected equally.